Hello, everybody. Welcome to the webinar today on harnessing the full potential of full spectrum cytometry, from panel design and acquisition to results in data exploration. My name's Sean Burke from Novo Software. We also have on the line Laurent Genestet from Cytek Biosciences, and we're really pleased to be able to communicate and walking you through some of the topics that we think are very interesting and relevant for the researchers out there today. So as a little bit of a background about the webinar today, part of this webinar is happening because Cytek Biosciences and Novo Software have this close partnership, essentially to seamlessly read raw spectral data in FCS Express and read the unmixed results that come off of any of the Cytek instruments. And as part of that, we've partnered to provide a 1-year license of FCS Express to Aurora owners. So if you've recently installed an Aurora, going back within the last year or two, most likely, you have access to FCS Express. So you can always reach out to us after the webinar if you aren't sure how to grab that or get on that license, but rest assured it's there for you. Now, in today's webinar, the full spectrum profiling technology on board the Cytek Aurora, the Northern Lights, and the Aurora CS systems really give researchers the ability to analyze more markers per cell and move in that kind of high dimensional data analysis space. And what the Cytek team is going to do today is come in and share their high complexity panel design principles and tools that are going to help you optimize the quality of the highly complex multicolor data. And then our team from Novo Software is going to come in and walk researchers through some analysis workflows in FCS Express for Cytek Aurora data sets. And essentially with FCS Express, we're going to move from a type of kind of standard, what you'd consider classical analysis, into a high dimensional data reduction, using tools like tSNE, uMAP, and FlowSOM. And we're really excited at the Novo Software side to be introducing pipelines today and some of the visualizations that are going to help you, better understand some of these data sets. So with that, I want to hand the presentation over to Laurent. Hi, Laurent. Welcome to the call today. We're going to make you a presenter. All right. And Laurent, I think we've got you on the line here. Are you there?
Yeah, thanks, Sean.
All right, excellent. Yeah, perfect. So I'm gonna let you take it away here. And again, keep those questions coming as the presentations go, and we'll make sure to get back to you. Thank you.
Yeah, thanks, Sean, for the intro. So as Sean said, my name is Laurent. I am the Global Marketing Product Manager here at Cytek. And I'll be going over our full spectrum approach to fluorescent flow cytometry and then some tools available to you when you're optimizing these multicolored data sets. Okay, so I'll start with just a very, very brief history of Cytek, and then we can dive into our technology and review those tools. So for those of you that have heard about Cytek or are joining from, from kind of our messaging, it's probably because of the Cytek Aurora Northern Lights or the newly launched Aurora cell sorter. These are now being used in a variety of different research areas, but Cytek actually used to be known as Cytek Development back when we were a third-party service provider for flow cytometry equipment. And then in 2015, that became Cytek. And then kind of throughout the years, the Aurora, the Northern Lights, the cell sorter very recently was just launched. And in all three of these cases, there is that full spectrum profiling technology built on board, which we'll go into in a few minutes. That technology is what enabled the publication of this 40-color uMAP paper. And then additionally, in that paper, we use some of these Cytek cFluor reagents, what I'll be talking about as well, which serve as an excellent starting off point as you explore kind of what the capabilities of what full spectrum cytometry has to offer. So a snapshot of where we are today, where, as Sean mentioned, we have attendees from all over the world. We also have kind of units placed across most of the, almost every continent with field service, applications, sales personnel scattered throughout that globe. We're trying to stay true to our roots of that kind of original customer support company, trying to help support your instrument and your research. And so what is full spectrum profiling, right? So this signature that you see here, if you can see my my cursor, this signature that you see here is key to understanding what full spectrum cytometry has to offer, and there's a lot of information kind of packed into this one signature. Let's start with on a conventional flow cytometer. This is how we would detect FITC, right? So the instrument would have specifically designed mirrors and filters to ensure that the detector, in this case shown here, measures the peak of, in this case, FITC. And then here's what we would call maybe a PE detector. And you're seeing some light here that we're having to compensate out this spillover. And then the rest of the data is essentially ignored signal from FITC, right? We're only interested, we've designed these filters and mirrors specifically to detect the maximal emission of FITC and nothing else. On a Cytek instrument, we would actually measure all the emitted light from FITC. So starting from the excitation signal or excitation wavelength of that blue laser, we have an array of detectors here from B1 all the way to B14, so 14 different detectors off of the blue laser that measure the entire spectrum of light, with some gaps here for the excitation wavelengths of, in this case, the yellow, green, and the red laser. And so here you can see we're going to start using all of that light. We have 5 spatially separated lasers. In this case, this is a 5-laser Aurora instrument, and that emitted light is then routed to the different APD detector modules via fiber optic cable. So 1 detector module for each laser. This enables us to capture the emitted light from each laser separately. We'll also have 1 forward scatter and 2 side scatter detectors, with the side scatter violet detector being especially useful for better resolution on small particles. And so each of these detector arrays are comprised of avalanche photodiodes, and then that is combined into a signature. We use avalanche photodiodes for a number of reasons. One, they're very small, so you can imagine on this particular configuration, we have 64 detectors that we want to fit in a benchtop instrument. And then second, they have high quantum efficiency across the entire region, but specifically in the far red regions. And so what quantum efficiency is essentially the ability of a detector to convert photons into electrons, and ultimately to convert that into an electronic signal, which you're seeing on your computer screen. So the higher quantum efficiency means that the detector can detect dimmer signals, especially, like I said, in this far red region. And so going back to the signature, which is a super important plot to understand, which contains a lot of different information about the fluorochrome. So on the y-axis, you will see the median intensity. And on the x-axis, you'll see every detector on the system. Again, this is on in this example, this is a 5-laser instrument, UV, violet, blue, yellow, green, and red. And the colored rectangles in each plot are, it represents 1 detector. It's a density distribution of the events measured by that detector. So here you can see the difference. This is how the Aurora would see a particular fluorochrome, with the peak emission from that fluorochrome being here in violet 14 and 15. And then just to kind of show you the difference, this is what a conventional instrument would see, with a detector specifically designed just to detect that information. So with a lot more information available to us, what are we doing with all of that? So many of you have probably seen this, the classic example of APC Alexa 647. We think of them as identical dyes in a lot of cases. And if you go to a spectral viewer, you'll see a lot of overlap here. So you would correctly conclude that you can't run these together. But looking at the full spectrum, of course, now we can see that there's a lot of differences in the full spectrum signature of APC versus Alexa 647. And this allows us to actually run these 2 dyes together in the same panel. So in this case, we're running them on co-express markers, CD8, CD56, and these 2. So this is a demonstration that even fluorochromes with highly overlapping signatures can be used effectively in combination. And what this means, from a broader perspective, is there's so many more fluorochromes available to you when designing your panels. So in this case, we've tested at this point over 90 dyes with many unique dyes across on every on the different lasers. So just as an example, on the violet laser, this shows 19 unique dyes with unique signatures. The other thing we can do by looking across the entire spectrum is start looking at autofluorescence. So here's 3 examples of different cell types: red blood cells, alveolar macrophages, and lymphocytes. And you can see the different not only the different signatures, but the different intensity of autofluorescent signal that we are measuring on the Aurora. And this, by measuring it, we can now extract that. So in this case, we're going to be looking at iPS cells transformed to express EGFP. When you're looking at the non-transformed versus the transformed cells, there's a very slight difference here. And by extracting the autofluorescence from these non-transformed cells, we can actually, with autofluorescence extraction, create a lot better separation here compared to without autofluorescence extraction. So again, just another way of using the information across the entire spectrum that's collected on Cytek's Aurora, Northern Lights, or Aurora cell sorter systems. Now going into kind of more practical applications of this was at the Institut Gustave Roussy Cancer Campus in France. And so they were looking at reliable ways to predict a patient's response to diseases like COVID-19 and then find treatment targets for that. And so in this case, this was a 25 color panel that was run on blood samples from each patient that was collected and then also analyzed via at a genetic level. And so this was able to kind of look at this calprotectin as a predictive protein to see whether a patient would have mild, moderate, or a severe reaction post-infection. Just kind of another example for infectious disease research. In immuno-oncology, using liposomes to target antigen-presenting cells, critical for cancer vaccine efficacy. In this case, it was a 24 color panel. And then I kind of highlighted this in one of the first slides, but then in September 2020 in that uMAP 69, looking at 40 different markers in a single tube, right, which was a huge leap forward in terms of the complexity and the kind of answers that we could derive from a single sample. And then also looking at that same data through high dimensional or dimensionality reduction algorithms, which Sean is going to touch on later about how you can do similar analysis in FCS Express. And so, as you can imagine, developing this kind of high complexity panel, we're still using the same basic principles of panel design. And so it required a lot of effort in terms of the original panel design, panel optimization, data troubleshooting to get to that point. And so to help researchers achieve their own assays of similar complexity, we're continuously releasing new tools to help with everything from the very beginning in panel design all the way through to data troubleshooting and data analysis. So I'm going to go over a few of those now in the next 10 minutes or so. So the very first tool you're going to see is actually the software on the Aurora or the Northern Lights. And so that's our SpectraFlow software. We spent a lot of effort making it both intuitive to new flow cytometrists and familiar to those more experienced with other acquisition software. And it really follows a similar workflow, but there are wizards that kind of step you through how to create your experiment. So when you create the experiment, choose the fluorochromes you want to run in your assay. You then run your labeled tubes, load them on one at a time, and run them to collect your single color controls. You unmix your data, which is essentially a fancy form of compensation. You unmix your data to look at the individual signatures. And then once that's done, the data coming off from your multicolor sample looks just like data from any other instrument, right? You're looking at live unmixed data in your analysis worksheet. And here's just another representation of that. The workflow is really no different than what you're used to on a conventional instrument, with some added benefits. So first, our instruments collect all the emitted light, right? We kind of went over that. So any dye excited by the lasers on board your instrument can be used. That means there's really no need to design your panel around the available detectors on your instrument, nor is there any need to swap out any filters or mirrors to accommodate your panel design. Second, all of the instruments are preloaded with these optimized Cytek assay settings. This removes the need to adjust voltages on individual detectors, which you can imagine is especially important when you're using like our 5-laser instrument with 64 detectors on board. So those 2 steps are removed. And then from then on, it's really similar, right? You're going to collect your unstained comp controls, which we're calling reference controls. You're going to calculate either on a conventional instrument, you'd call it compensation. On the Aurora or the Northern Lights, you call it unmixing using our wizard. And then the data coming off of it, again, looks, right, the data coming off of it looks like you would expect from any other instrument. Taking this one step further, you can actually take those signatures, which we discussed earlier, and store them into a reference library. And then you can reuse those, assuming that your fluorochrome and your instruments are stable. You can reuse those over time. And really, you can kind of skip these steps of having to acquire new reference controls every time, again, assuming the reagents and the instruments are both stable. And so we tested this using a lyophilized samples where we ran the reference controls on day 0, and then we just kept using those same controls over time, just running new sample every time. And you can see, looking down column by column, that the data looks the same from day 0 to day 77. So this is all data this is an instrument that was passing daily QC every day, right? And so the controls are also being normalized against that daily QC. And this is, like I said, from a lyophilized demo kit that we use. So in addition, both in our software and online, we actually have spectrum viewers, which provide indices like the similarity index or the complexity index. And these are quick ways to determine, so the similarity index, starting with that one, it's a quick way to determine how similar 2 dyes are to one another when looking across the entire signature. And the lower the number, the more dissimilar those 2 dyes are, so the easier they will be, the easier it will be for you to combine these 2 dyes together in a panel. And then the complexity index is a measure of the overall panel. And so you're looking at kind of overall across, let's say, a 16-color panel, how do those fluorochromes interact with each other? So like I said, it's a bit, you can see these indices directly in SpectraFlow, and then also on our website, there is a link to the spectrum viewer where you can kind of load these in and see how do these dyes perform together? How do you expect these dyes to perform together in an assay? And then finally, using all of this knowledge, right, from our 40-color panel to kind of new matrices to determine fluorochrome similarity and panel complexity, we've started offering now new kit solutions using our cFluor reagents. So this is an example of our 14-color kit in this example, but we're doing the panel design and optimization and the individual reagent titrations kind of for you, right? So that's saving you time and effort. We take into account antigen density, stain index, spillover, making sure that final population ultimately can be easily identified. So we do all the reagent titration. We provide a recommended protocol for preparing your samples as well as for acquiring your samples. It comes with an experiment template and analysis worksheet. And ultimately, what we're trying to do is set you up for success when running your full samples, right? With pre-built gating templates to look at the major cell subsets. So this is kind of the next step for us in terms of empowering full spectrum research. We have 2 kits available right now, an 8-color TB MNK kit and a 14-color immunoprofiling kit, which you can see here. And so the goal here is to kind of jumpstart that research, to jumpstart that full spectrum research, right? Give you a really good foundation on which you can build upon and expand on these panels to get to your higher complexity. But this is a great way to see kind of what good, high quality 14-color panel would look like. Both of these are only using, as you can see, violet, blue, and red dyes. So like I said, expansions are possible if you have additional lasers, or even if you want to add more dyes in off one of these lasers. And so finally, just kind of summing up benefits of the full spectrum profiling approach. we are capturing the entire emission across multiple detectors. So a lot more data is being collected from each fluorochrome. And using that data, what we can do is provide additional flexibility in these fluorochrome choices. So I talked about APCLS at 647. This is one of our Cytek cFluor reagents, which you can run together with PE. But again, on a conventional instrument, these would probably occupy the same detector. So a lot more flexibility in your fluorochrome choice. You can use more fluorochromes together in a panel. Ultimately, that is providing you the ability to increase the complexity of your assays and run and drive at more answers per sample. We also have, kind of along the same lines, taking assays that maybe required 3 tubes before and combining all of those markers into a single tube, which is especially important when you're working with a limited volume of sample. And then finally, I touched on autofluorescence extraction samples like lung tissue or tumor samples we've seen with high autofluorescence, and now the ability to characterize that autofluorescence and then extract it is possible now with this full spectrum profiling technology. And so with that, I think I'm going to kick it back over to Sean, where we can talk about how we're looking at these panels, right? And we're able to take that data from the Aurora Northern Lights or Aurora cell sorter and bring it in a streamlined way, bring that over for analysis into FCS Express.
Excellent, thanks, Laurent. And I'm gonna flip this over to myself here, and let's make sure that we have that coming through. And Laurent, you can see the screen coming through here, right?
Yeah, I can.
Excellent. Yeah, so thanks so much. And I see that we have a bunch of questions coming through the questions and answers panel, all really, really great questions. We're going to make sure to try and get some of those addressed in this portion of the webinar and come back around for a little bit of discussion on those at the end as well. So with that, we're kind of making this transition from acquisition and moving from the instrument and spectral flow to starting to get some more kind of final results. And where we want to do here is show you how we can utilize FCS Express in some of the workflows that we've developed in the software to really help you get to that final result from your data. So one of the things that we're going to be covering here is utilizing pre-built analysis panels for the Cytek 14-color cFluor kits. We're going to talk about kind of expanding that into more custom-built panels. The use case application there is going to be a 35-color COVID panel kit, so really kind of making that jump to more than double the number of markers and colors in there. And we're also going to come back and discuss utilizing pipelines in FCS Express to help move from a standard type of analysis, say two-dimensional plots looking at, parameter 1 versus parameter 2, to this high-dimensional data reduction, getting into using algorithms like uMAP and FlowSOM. But really, I think one of the important things you're going to see today is that utilizing a pre-built panel is a really great way to serve as a backbone for high-dimensional data reduction. And again, implementing the pipelines in FCS Express to arrive at uMAP and FlowSOM, it's very easy, but it's made easier by having that kind of standard analysis to kind of fall back on to help you identify and name and work with the phenotypes of interest. And we're going to come back around and also show you in FCS Express, how to utilize some of the high dimensional data reductions visualizations and how to interpret them. So with that, let me kind of come out of FCS Express here, and we're going to jump, come out of PowerPoint, and we're going to jump into FCS Express. Now, what you see on the screen here, this is not PowerPoint, right? I know it kind of looks very much like Microsoft PowerPoint, but this is FCS Express. And the kind of whole point of that is you've worked in PowerPoint a million times, you know how to use this sort of thing. Now, the template that we're working on here, I'm going to show you where you can kind of go to get these. This is available from the Novo Software website. You can come to the website, you can search for Cytek in the little search bar here. And if you scroll down, you're going to see some example analysis and templates like the 40-color analysis template that Laurent mentioned, the COVID-19 35 color immunophenotyping panel, and as well as this 14 color kit we're about to dive into. And you can download these, right? And you can kind of pick up, where you left off from SpectraFlow and from the Cytek instrument here in FCS Express. Now, the way that these kind of templates are arranged is, there's some information on this first kind of page or the slide of the analysis. We come in and we show you a little bit about what the analysis strategy is supposed to be here, the subsets that we're looking to identify. And then we have many pages that are kind of devoted to the actual analysis. And what you're going to see is that these pages are all blank right now, right? We haven't actually loaded up any data. But the way that we get that data into FCS Express is we can come over, to where we have the unmixed data or even, mixed data or raw data, and we can drag these data files into what we call the data list over here in FCS Express. And when we drop that, you're going to see that all of a sudden, all the plots update, right? They're filling out with this data that we've dragged into the data list. There's some little steps here on the left that kind of guide you through that process, but all of the analysis on all of the pages is now ready for you to come in and review and to look at. Now, generally, the first thing you want to do is take a look at your gates, right? You want to kind of move between the different data files, which we can accommodate here by utilizing the data list in FCS Express. And, one kind of thing that we can see here is we might need a small adjustment for sample 2 for this monocytes gate. Now we make this very easy. You hold down the shift key and you move that gate and it becomes what we call data specific for that file in FCS Express, while all the other data files here are using the same gating position. But what you might not have noticed here is that as I am adjusting this gate, everything in this layout is updating in real time. So that plot below where we're looking at the monocytes, even the spectral overlay chart that we're showing here, mainly as an example to give you an idea of, you can work with the spectral component of this data set, but all of this is updating in real time, right? So essentially you can pick up where you left off, get your data into here, get this live updating kind of feedback and results. So, even, our histogram overlays, as we kind of go through this analysis, you have an idea of what's going on. Now, an additional item that's built into this kind of pre-built panel is on the CD3 positive page here, where we're looking at CD4, CD8 ratios. This is not intended for IVD use, right? If you don't need these ratios, you can always come in here and remove this, but it's a pretty cool thing in FCS Express that we can enter in just in these little kind of fill-in or edit boxes, some sort of range. And what will happen is as we move between data files, you can see here that CD4, CD8 ratio is being automatically calculated. And if we go back to this first data file where things are out of range or more than, the 5 that I manually entered here, you get a little warning, right? So there's a lot of quality control checks that you can build into these analysis templates. We've built this in as a, just a bit of a control so you can see how that works, but it's very easy to kind of work with. Now, additionally, when we talk about a final result in FCS Express, what we mean is something that you get ready to publish, something that you bring to your lab presentation that week or that day, integrated directly into the software, something like this bar chart. Now, with this template that we've created, you can see that we've looked at CD4 and CD8, the effector memory, the naive cells, central memory, effector memory, right? And it all links up to this quadrant, right? So as I move these quadrants, you can kind of see on your side that these bars and these numbers are all updating in real time. Also, when we look over here in our CD8 positives, right, we get that live updating information and feedback. So again, by utilizing the charts within FCS Express, which are rather easy to create, you can get this live updating feedback. And that also goes for things like pie charts, different styles, regression plots. And again, as we navigate through different data files here, all of that information is going to update. Now, in the end, we know that you do want to get this data out, right? You're generally going to want to go to PowerPoint or to PDF or to Excel. So what we've done in this pre-built analysis template is we've built in what we call batch actions to essentially export this data to PowerPoint into Excel automatically. And what we're doing here now is we have 3 files in the data list, and we're performing 2 actions, exporting to PowerPoint and exporting to Excel. And again, all we've done with this analysis template is load in our data files, adjust the gates so they're in the positions that we need them to be, and click run. And just like that, Excel has launched up with all the reportables that we're interested in. On the back side here, I should have PowerPoint launching up. So let me see if I can minimize some of this stuff and not get too confused where I'm at. Right, here we are in PowerPoint. And what you can see in these PowerPoint documents is that all of these plots are high resolution individual graphics, right? So if you need to look at the CD3 positive and CD3 negative, you can blow this up, you can get it ready for your lab presentation, your poster, whatever it is you might need. So again, working with these templates is a great way to kind of pick up where you left off in SpectraFlow. But what happens if we get more colors, right? We come in here and now we want to get into this kind of even higher dimensional space. And I'm going to pull up the 35 color COVID panel that we had worked on a little bit. So again, what we've done in this kind of panel, and again, you can create these on your own, is we've built out, all of the gating hierarchy that we need. We've built out the main subsets. We've put in charts showing absolute counts here. We've broken it down into monocytes, classicals, intermediates, non-classicals. And you can see that even though this is a very, very, large 35 color panel with some very complex results, by templating this into some sort of format, it's going to allow you to make a lot more sense of this data a lot more quickly. And you might be wondering, all right, well, how do I set this up on my own? I didn't run the same exact 35 color panel, right? Well, when you're working in FCS Express, what you need to do is think about, what you might do in PowerPoint. And there's a lot of drag and drop in PowerPoint, and there's a lot of drag and drop in FCS Express. So for instance, if I wanted to start kind of building up my own analysis here, I can drag the singlets gate out and I can drop it onto the layout here. And what that's going to do is it's going to open up a new chart in FCS Express that has that singlets gate applied. So once it gets done opening up this chart, essentially what we're going to be able to do is drag and drop and drill into all of the populations of interest that we want to navigate to here, right? So again, when we are working in FCS Express, don't think about what you would do in other cytometry software programs. Think about what you're going to do in Microsoft PowerPoint to insert objects, to insert tables, to kind of allow you to walk through all of this data and create these analysis templates for your own. So again, I do want to point out that, over on the website here, we do have the 35 color immunophenotyping panel. This can be a backbone for a lot of the things that you're doing. We have the 14 color panel here that can be, again, used as a backbone. We have some other panels here, and of course, going in and exploring the 40 color data set there. So FCS Express, again, we're back here. We've opened up that chart where we're looking at the singlets here. And again, if we want to start building this out, you can drag and drop that singlets gate out, and then you can pick and choose, right? We've got a lot of parameters here, so we'll change the side scatter area versus live dead, right? And then we can come in and create a gate on this. And again, FCS Express will create that gate. We can drill down into the live population and then go in here and look at CD45, right? Drill down into that population, kind of building up this analysis template as we want. But we do understand, right? This is very high dimensional, very complex data. And as good as this layout design is, as organized as it is, it's also helpful to have displays and visualizations that are going to help us distill this really complex data into a very simple visualization, something like this, right? And I'm going to kind of make this a little bit bigger and zoom in on it a little bit. And just like PowerPoint, we'll bring this to the front here so we can see. And what we're looking at here is 4 different overlays for different gated populations. And then we're looking across 14 parameters here, right? So for CD8 TEMRAs, we're getting the heat of all the different parameters for those populations. Same for naive and central memory and effector memory. And these sorts of visualizations, again, are helping us distill, 4 or 5 different gated populations or samples across all the parameters. So how do we build this up? Because this is kind of a transition to moving into algorithmic type high dimensional data reduction. Well, what we do is we can insert something like a parameter heat map, and this parameter heat map is going to display with some sort of default. Okay, now this default, we need to make some changes to facilitate the type of display that we want to work with. And what I'm going to do is I'm going to right click on this and choose format. And we're going to have all of these formatting options, very much like you would use a Microsoft Office. Now, the first one I'm going to want to come in and take a look at is I want to change this from landscape to portrait, right? And I want to kind of put my sample up on the top or my gate on the top and run the parameters down the side. Okay, now, in this case, I have one sample or one gated population, but if I want to compare a few different gated populations, what I can do is I can just drag and drop a plot or drag and drop a gate, and I can say add it as a new overlay. You're going to see it creates another kind of column here for that overlay. And I can do another one here, right? I'll just do 3 today as a very basic example. Now it adds these in here, but what we really want to do is be able to come into our overlays and say, all right, well, for the first overlay, let's come in and look at CD8 naives, right? Now all of a sudden, some of the heat mapping starts to fill out, right? For the second one, let's look at CD8 effector memory, right? For the third overlay, let's come in and look at CD8 central memory. And just like that, we've kind of built up this heat map very similar to what we have here. But of course, we want a little bit more formatting, right? We can put in the gate at the top and it's going to show us the gated populations. Also, kind of very importantly, there's a lot of color level options here. So I think looking at these with kind of a red, white, and blue scale is very effective. But also, because this is a 35 color panel, we may or may not want to look at every parameter there, right? So what we can do is come in and specifically tell FCS Express, all right, let's visualize CD16, CCR5, CD11C, CD56, CD8, CCR7, right? We can kind of go through this list and build up this kind of high dimensional matrix visualization any way that we want. So again, what you've seen here in about 2 minutes of me kind of explaining over this as we're doing it, we've built up a high dimensional visualization where we're looking across 13, 14 different parameters. We're looking across 3 different gated populations, and we very easily could have added additional samples, additional gates to kind of build this up any way that we want, and we have it in the format that we need for publication. Okay, so again, this is kind of an in-between, moving from kind of lower dimensional, you're moving into a higher dimensional space now. These visualizations are still based off of standard gating and visualizations and things like that. But how do we kind of graduate from that into something like high dimensional data reduction tools such as uMAP and tools such as FlowSOM, integrating things like high dimensional data reduction tools like Flow AI and FlowCut and things like that, right? So what I'm going to pull up here in just a moment is some uMAP charts, all right? And we're going to be visualizing some of this data in uMAP. We're going to be visualizing some of this data in FlowSOM charts. And what you're going to see in FCS Express is that when we work with these tools within the software, they're all natively implemented. Now, what does that mean to be natively implemented? That means that the software tools are going to be running on your local computer, utilizing the kind of RAM and processors and resources that you have. It means that we're not going to rely on R. Right, so very importantly, you're not going to have to be a R scripter. You're not going to have to be an R programmer. You're not going to have to be a biostatistician to kind of put these things together, like you see up on the screen here right now. Right, so the way that we access tools like uMAP here are through our transformations icon here in the Tools tab, and you can see that we've set up something called a pipeline. And really, what a pipeline is doing is it's a set of steps that we're going to perform some sort of action. So in this pipeline, we're going to do an interval downsampling, right? We're going to run uMAP on 100,000 events, and then we're going to run the uMAP algorithm on this data, right, to arrive at these plots. And just so you kind of see how easy this is, right, we create a pipeline. It creates what we call a root step, where you essentially just pick all of the parameters that you want to perform in this transformation. And then we come over here and we say, hey, well, if you want to run FlowCut or Flow AI on this, make that the first step. And this is a predefined algorithm, meaning that there's 3 steps in here that we've already put together for you rather than you having to figure that out on your own. And then we come in and we say, hey, all right, well, on that Flow AI clean data, let's do an interval downsampling on that, right? We'll kind of pop this into the correct location there through a drag and drop. Now we're going to downsample to 1,000 events. Now we're going to come in and we're going to do, a principal component, a tSNE, a uMAP, right? Whatever it is that we want, kind of put that into the pipeline here. So when we apply this to a plot, it's going to run Flow AI, it's going to interval downsample, it's going to run uMAP based off of the default steps. Now, this is all point and click kind of interface, right? So if I change from, working with 100,000 events and I say, let's just run 1,000 events for this uMAP, well, when you do that, FCS Express is going to calculate on the fly, and you can see that our uMAP charts over here to the right have updated, right? And if we change that variable to something like 5,000, the uMAP charts are going to recalculate. And again, you can see how fast we're actually doing this. I know the downsampling is quite low here, but again, you come in, point and click, you don't know what these variables do, but I'm sure you can change them, right? And you can come in and see how this is impacting your data. Now, we kind of understand a little bit about what these pipelines are doing, but how do we kind of implement this and kind of get some more displays and start making sense out of what this data is actually telling us, right? Coming in and distilling some of that high dimensional information. So let's close out of this layout. We'll come into a slightly modified version of this. But again, what you can see is I'm working on the backbone here of what we had already put together. Let's expand all of these, all of the gates, all of the main subsets, everything that we've kind of put together for this kind of standard analysis. We're now working here in a more high dimensional mode where we're going to be working with uMAP, and we're going to be working with FlowSOM, and we're going to be doing consensus clustering. And don't worry, if these things don't sound familiar to you, they're very easy to implement. What you're going to see here is that when we create something like a uMAP chart like this, and again, so you can look at the transformations and the pipelines, we have these uMAP steps here. Well, when you get those charts, which you can kind of see off to the right on my sandbox page here, you still have to come in and kind of identify and name what these clusters are, right? Now, one of the really important things about having this kind of standard backbone, for the standard analysis is, well, we've already done a lot of gating, right? We already have identified, all the major subsets that we need to look at in our gate view. So what we can kind of accomplish now is with this kind of 2-dimensional plot for uMAP 1 and 2, well, we can come in and we can turn on and off backgating, right? So if we wanted to come in and say, hey, I wonder where the B cells are in that chart, well, let's turn on the B cells population. And sure enough, this little kind of population in the bottom left is highlighting because that's our B cells. And then if we come over to our gated data from our standard analysis and we move that gate, you're going to see that that population is updating based off of that. Right? So, again, this is kind of a classical way to come in and do some of this phenotype naming, is use the kind of gated populations that you already have, use things like backgating, which we can accomplish here with the gates to display, turn these things on and off and try and figure out, where all of these populations are, give them some sort of name. And then when we need to do comparisons between samples, we know what these populations are. Right? And then when we come over to uMAP visualization and we kind of look at this data across all the different parameters for this data set, well, we know this kind of population on the bottom left was our B cells, right? And then we can come through and kind of confirm that, right? We can look and see, are they CD19 positive? Are they CD20 positive? And we can determine all of that by the heat of these uMAP charts that we've inserted and that we've worked with, right? So again, this is helping us with the data exploration, right? It's not coming in here and automatically identifying the clusters and automatically naming them. We are having to use some manual input here about what they are. But once we have that, we can come over to our visualizations and we can say, all right, what does this look like at a kind of panel, like, kind of global level? If we have multiple data files that we've merged together, we can deconvolve them and then look at our uMAP charts side by side, and we can look for the changes in heat between the different individual parameters and kind of utilize that. But, a lot of folks are now starting to use tools like FlowSOM. So what FlowSOM is going to be able to do for us here in our transformations, you can see that we've inserted it here into our pipeline routine here, or our pipeline step. And FlowSOM is a predefined algorithm for us, right? So you don't have to know how to put all of these steps together. But essentially what we're gonna do with FlowSOM is we're gonna look at uMAP 1 and 2 parameters for the uMAP we've already defined, and we're going to try and cluster these clusters up here, right? We're going to try and algorithmically define where these clusters are, rather than having to go back and try and manually define them. And that's what our FlowSOM is doing down here. And it does this by, in this case, we assigned it to 8 different clusters for the 35 color panel. There's probably many, many more clusters that we could have identified, but for the sake of simplicity, what we've done here is consolidated this into essentially 8 different clusters. Now, you still have to create gates from these clusters, and you still have to get these kind of applied to different plots, and identify and name what these populations are. But with the new tools in FCS Express, we can create what we call a well gate around this. And I'll bring this dialog over here, and we can say, hey, well, for all the individual clusters, create a gate for them. And what FCS Express is going to do, and you can kind of see it up here in the gate view, is that it's created 8 different classification gates here, right? So what we can do then is take that gate and apply it to a uMAP and say, hey, there's cluster 1 that FlowSOM identified. Here's cluster 2 that FlowSOM identified. Here's cluster 3, 4 that FlowSOM identified, right? And then we can come back in and we can start, looking at this data, trying to figure out, what these clusters actually represent, what they actually do, what they relate to, give them some names, and then go back and start kind of parsing out all of that information to arrive at some results. Now, when you're generally done, working in that sandbox mode, you're going to want to come in and, I say sandbox mode, I just called that my sandbox page. It's kind of my data exploration page for me to kind of put together some results. Well, we can put all of this together in FCS Express so it's ready for publication, right? So we've taken that FlowSOM chart, we've kind of changed the color schemes, we've looked at CD4 as the heat here, so we can kind of see that these clusters in red towards the top of this FlowSOM chart are CD4 positive. And if we come in here and we look for CD8, we change what's on that axis, right? Things branching off here to the right are CD8 positive. And again, we can take, that uMAP chart, we can, add some different backgrounds to it, change the color schemes on it to make it look really, really sharp. But again, the other way that you can come in and distill this information is getting back into those parameter heat maps, right? So just like we did with our standard data before, what we've done is created multiple overlays in this chart, right? And those overlays are just simply the FlowSOM defined meta clusters, right? We called this FlowSOM CD4 positive, FlowSOM CD8 positive, CD3 negative B cells. And then we've listed all 34 or 35 parameters in this data set on the Y. And just like that, we now have a visualization where we can come in and say at a very high level, what's going on with this data set between all the different parameters, all the different gated populations. And again, when you're ready to take this and you're ready to move into some sort of other format, we can come in and very quickly export to PDF, export to PowerPoint, export as a high resolution TIFF. And in this case, I'll just kind of export this page so you get a very quick idea of what this looks like in PowerPoint. And I believe PowerPoint's going to be launching up on the other side for me here. So let me go over and grab that. And here we are in PowerPoint, right? Again, there's our uMAP. We're ready to put this in our poster. We're ready to put that in our publication. Here is our, parameter heat map that we put together, right? Here's our FlowSOM, and we have everything ready to go, ready to move out to publication ready format, okay? So again, I know there's a lot that we went through here today, right? And there's only kind of so much that we can cover in, 20, 25 minutes, right? We can't cover a full like end-to-end how to do, uMAP and FlowSOM. However, we do have a lot of webinars and short videos that are currently available and more coming. But I think what you can see here is that FCS Express allows you to easily move from that full spectrum or full spectral profiling data sets from the Aurora and Cytek systems and SpectraFlow and get it into a format that you're able to report on those results very easily. The pre-built analysis templates are a great tool to get started with off-the-shelf kits, right? So if you have the 14 color kit, if you've developed your own analysis panel, because creating and designing those custom templates is easy to do, that's really also going to help you set up for success with the high dimensional data reduction and the exploration. Because by having all of those gated populations and being able to, run your pipelines on top of the data and the gates that are already there, you can link up, right? You don't have to go back and kind of re-identify and rename. You're already, halfway there to that high dimensional data reduction. And also the ability to use the pipelines in FCS Express makes these tools very easy to use. They're very fast, they're native, you don't have to rely on other software packages. And because it's in an intuitive interface, it becomes very, very easy to use and work with. So with that, I know we've covered a lot today. Again, moving from the full spectrum cytometry panel design and acquisition, covering and moving out to some results in the data exploration in FCS Express. And with that, I want to say a big thank you for everybody for attending this webinar. I also want to give a big thank you to Laurent and the teams at Cytek. I know we had a lot of folks kind of working on the background with these data sets and putting together analysis templates, including Maria at Cytek, including Kate Pilkington at Cytek, and many others, as well as our team of experts at Novo Software. Big thanks to Robin Kamachika, Kea, Andrea Valle for working on the back end with some of this data so we can present it and putting together these pre-built panels for folks to use. So with that, we're going to pause for a moment and we're gonna take some questions here. So thanks again for attending. And again, we're just gonna take a few questions.